Hi everyone, I'm Kenny Travouillon. I'm the curator of mammals at the WM Museum. And I'd like to acknowledge that I'm presenting on traditional lands of Whadjuk Noongar people and pay my respects to the leaders past, present, and emerging. I'll be presenting on our work on the golden bandicoot taxonomy that I'm doing in collaboration with Terry Bertozzi, Steve Cooper, and Anna MacDonald. Now, the golden bandicoot is a threatened species, and here you can see on the screen the past distribution in white, with the white circles here, of the species. And the current distribution is, you got a subspecies on Barrow Island, Barrowensis, a population in Kimberley, which has been assigned to Oratus, and then the population on Marching Bar Island has been assigned to Anamensis. But I'll come back to that, why I don't think that's the case. So if we're looking at the history of the taxonomy of this species, it's been actually pretty difficult to untangle what has been done. The Mammal Society this year actually put some guidelines on how to tackle taxonomy in mammals, and they suggested that papers tackling taxonomy should definitely look at types. They should have molecular data, morphometric data, have a broad geographic coverage, and have descriptions. So when we're reviewing all the papers that have tackled the taxonomy of that species over the years, not many of them actually would tick boxes in all of these categories. So the early descriptions obviously wouldn't have any molecular data and a very, very small coverage. A lot of the works that made decisions on the taxonomy actually are checklists, and often they don't present any scientific work towards that their decision making. So there's no way you can actually check why they've made that decisions. Probably the best work so far has been the Lin and Mortz in 1981, where they actually did look at types, morphometrics, geometric coverage, but that was before genetics even started. And then more recently, all the work has been on the genetics, where morphology hasn't been looked at in those papers. So there's a disconnect between the morphology and the genetics. If we look at specifically actually of the geographic part of it, I've here separated Oratus, the Kimberley, from the inland, with being the proper Oratus being the inland, and same with the Anamensis, being from Annamanland rather than Marching Bar. Again, most studies didn't really look at everything, except from this one at everything but Marching Bar, and the genetic studies at everything except the two extinct populations. So there's no one really ticked every boxes, and this is what we're hoping to do in this work. Just to give you a bit of background of what the genetic work has been done so far. So that was published a couple of years ago by my colleague Steve and his colleagues. And this is mitochondrial DNA. And golden bandicoots are appearing all over the tree, so they're polyphyletic. And so that's been a bit of a problem, is that, is this one species or multiple species? And it's been bothering a lot of people trying to do conservation on this species. So in our study, we looked at all the types. We've looked at almost every single specimen in existence, so all 212 specimens in museum collections all around the world. And we collected data on skins measurements, skull measurements, dental. Through the OMG project, we got tissue samples, which represent 2,000 nucleogene exons, and that's 105 specimens within the genus Assurodont, including some of those old museum skins, which are very hard to get a DNA sequence from. For the morphological data, it's been analyzed with PCA, discriminant function analysis, and MANOVA. And we're also hoping to do some field work next year in the Kimberley to add more. So quickly going through the skull morphology, and I'll be very brief about this. So these are the skulls of the three types. And you can see here, they all actually have the same scale. That's the one at the top is the smallest, and that's the inland golden bandicoot. And surprisingly, it's actually smaller than the Barrow Island golden bandicoot, which would most people would expect the island species to be smaller. And the unnamed one was the biggest. And if you introduce the two extant population from the Kimberley and the Marching Bar Islands, Marching Bar is similar in size to actually that inland golden bandicoot, and then Kimberley is closer to the unnamed one in terms of size. So when we're analyzing the morphology of it, it looks a little bit messy in the PCA because there's a lot of overlap, but it's actually every population is actually distinct as a distinct. So the MANOVA is finding that the p-value is 0, so all distinct from one another, and it's more clearly visible in the discriminant function analysis. And I've added in the data set another species, Peninsulae, which is from Cape York, just to show how different another species is to all of these populations. So it is equally distant. The Barrow Island seems to be the most distant to all of them. And done a pairwise comparison as well for true orators versus the rest. And there's still quite a bit of overlap according to the pairwise comparison. So these are raw results. So what happens if we size correct? So meaning we are looking at any effects from being basically shrinking in size due to being on islands, and we get quite similar results. In the PCA, it's a bit more overlapping, but here what we see in the DFA is the unnamed land and Kimberley actually coming on top of each other, then actually not as different in that respect. And when doing the comparison from the golden Bandicoot oratus barrowensis, it becomes the most different one compared to the other two in here. With the bulle size, so in mammals, the bulle is basically that bone that is around the ear bones, and it will actually act as to amplify sounds. So species in the deserts tend to have a big bulla, while species in forest environments tend to have a small bulla. So I calculated basically a bulla index for all of them. So we have our golden bandicoot from the desert has the largest one, which is what we would expect. And we found like the smallest ones is from Barrow Island and Marching Bar. And the closest one will be unnamed land and Kimberley. So that's something to keep in mind for later on. So moving on to the molecular data now. And this is a very, very preliminary tree. So this has actually been cleaned up and checked yet for taking out all the poor samples. So things might change when this is done. But at the moment, here's what it looks like. So it's very similar to the mitochondrial DNA analysis, where we're recovering the population as polyphyletic. But we shouldn't pay too much we shouldn't try to work out really our species at this point, because there's still it's still too early to interpret that data. But if we compare the morphology data set, barrowensis is the one that still is sticking out away from the rest. So the conclusions, so the morphological data sets suggest that each of the different population is distinct, whether it's species or subspecies that will come up when we get the final molecular data sets. Marching Bar population should be probably considered a distinct taxon and probably subspecies. Barrow Island looks quite distinct, but we'll see what the final results tell us before making a decision on that. And Kimberley has been a bit of a problem because, as you saw, sometimes it goes with anamensis, sometimes with oratus. It goes all the way. So there's no, I really need to get the final molecular data to actually make a call on this, because I don't know at this stage whether to call it a subspecies, a species, or just a different variation of a population. So this will come later. But this is pretty important to make a decision on that very soon, because there's a lot of conservation work being done at the moment where people want to mix those populations. But I'd say it's too early to make that call, and because we are likely to have problems where these could be taxonomic entities that are quite unique and different, and like mixing them might be an issue. But the bullet size being an important part of the morphology, if you were to translocate one in the position of the extinct inland population, probably the ones from the Kimberley are the best ones to translocate at this stage. Thank you very much. If you have any questions, I'll be happy to answer them.
Thanks so much, Kenny. We've got plenty of time for questions. So if anyone has a question for Kenny, please feel free to raise your Zoom hand or write it in the chat.
Maybe not.
I think it's really interesting having a session where we've got entomological, decisions being made about species and cryptic species, and the same similar thing happening in mammals, where from your data set, it seems like you're needing a lot more data to kind of make those conclusions, and the implications for conservation can be fairly significant. How much more data do you think you'll need before you'll be able to make a decision? And does that decision kind of need to be a collaborative process, or is it something that you can decide and you think the conservation applied researchers will take on board?
I think at this stage we have the data, well, the best we can, because it's really hard to get the DNA sequences out of old skins in the collection. So the tree we have at the moment just needs to be cleaned up, checked, making sure that any of the poor sequences that are creating problems in the tree are removed, so then we can have a really strong tree. And then we can make a decision. So I think we're only a few months away of that work being finished. So then we can start making a decision and actually writing a paper, making a final taxonomic decision. And that should inform conservation down the track.
Awesome, thanks, Kenny.